Hello, welcome to the next episode of a CNA's Opus. My name is Rich. I'm a certified nursing assistant. Before I start, there's really something that I want to get out of the way, something that's been really bothering me, creating issues in my life, and I'm getting a lot of questions from people, and I really just want to put an end to it. So I have some things on a board here just to graph some things out. But first things first. First things first. I want to get this out of the way. My relationship with Sasha Grey is private. We're friends. We're friends. That's it. Done. Over. I don't want to hear any more questions. Okay, now we can move on from there. So, yesterday I was talking about the P53 pathway, and I'm going to continue that discussion with the P53 pathway, except I want to start from the very beginning. So, let's say that you suffer some DNA damage. And if you look at the board here, I ran out of board space, and there we go. So say there's a double strand break right there, and you have some DNA damage from the UV rays of the sun or some other mutagen, whatever caused it. And you have these two separate enzymes which have very awesome names. In fact, if I had a kid, I would name him T-longectasia because I think it's just an awesome name. But anyway, the two enzymes are ataxia T-longectasia mutated, and the other one is ataxia T-longectasia mutated Rad3 protein. So those are the two enzymes that are going around the double helix and checking for errors. And once they hit an error, it's going to activate them. And they're serine threonine kinases, which means they're enzymes that are going to pick up a phosphate group and bring it to another enzyme, and it's going to activate them. So it gets activated, it picks up a phosphate group, and it goes and it phosphorylates serine or threonine on a different enzyme. And as long as the functional group of the serine or threonine enzyme is exposed from the enzyme, it can do it. And I could show you a little bit of the chemistry right here. If you could see it right here, I know this board is really small, but over here is threonine and over here is serine. They're basically the same, except this one has 3 hydrogens. See, we have the amino group that's up top here. That's your amino group. And then the first carbon right there, that's your alpha carbon. And down here we'll have the carboxylic acid. And then off the carboxylic acid, we have an oxygen and a hydrogen. And over here, you'll see the difference. There's a methyl group right here, and then a hydroxyl group right there. The same thing occurs over here, except this one has 3 hydrogens off the carbon right there. So the same thing is going to happen because this one has a hydroxyl group right there, and this one has a hydroxyl group right there. So this phosphate group right here has the it'll bind, this hydroxyl group here off the phosphate group will bind with the hydrogen off the serine or the threonine over here, and it'll produce water. So it's a condensation reaction because it's producing water. And then the phosphorus atom in the middle is going to covalently bond to the oxygen of either the serine or the threonine. Okay, and from there, what happens is, now that we went over that, is ataxia T-longectasia. We'll just call them ATM or ATR because they become a tongue twister. So those are two separate enzymes that are checking for errors. And what they do is they'll go down, and using that chemistry that I just showed you, they go down to two separate other enzymes, which are checkpoint kinase enzyme 1 or checkpoint kinase enzyme 2. And that's the second step. And those are two separate enzymes. They're not dimerized, nothing like that. They're two separate entities. And what they do is that's the second step in the P53 pathway. What they will do is they will go over and phosphorylate P53. And when it phosphorylates P53, basically what it does is it protects it from ubiquinization. There's a protein called MBM2, which will bind to P53, and it'll inactivate it. So P53 is constantly being made in the cell. MDM2 will bind to it and inactivate it, inhibit it, stop its function, right? But it also targets it for ubiquination. So ubiquination, there's usually a chain or a tail that's formed. It's called like a polyubiquitin chain or a polyubiquitin tail. And basically, what it is like there's a lysine residue off of position 48, and it'll bind the carboxylic acid will bind there, all the way down to make this chain, and it'll pull in, take in the P53 and chop it up and destroy P53. So it's constantly being made and it's constantly being destroyed, and so there's damage in the DNA, and it phosphorylates it and it protects it from MDM2. So not only does it protect it from just stopping its function, but it protects it from being ubiquinized or destroyed. So once that P53 is phosphorylated, it forms this tetramer. 4 P53 proteins come together and form a transcription factor. The transcription factor will go down to the promoter region in the DNA, and it'll go to different ones. First, it'll go to ones that are required for DNA repair, because that's the idea, is to repair the DNA so that when the cell divides, it doesn't replicate, divide, and have damaged DNA in the daughter cells. So the first thing it wants to do is it wants to repair it. But if it doesn't want to repair it, it has other things like P21, which I talked about in the last video, which gets kind of confusing. But that is a tumor suppressant protein, so it'll halt the cell cycle. That transcription, P53 transcription factor, will halt the cell cycle. Actually, throughout the cell cycle, it could halt it. But what I want to talk about right now is the pro-apoptotic proteins that P53 will create. And we'll start off by it forms that transcription factor, and the P53 builds and builds and builds and builds and builds and builds, and it's not repairing that DNA. And eventually, what happens is it forms that tetramer, that transcription factor, and it goes down to the promoter region. And RNA polymerase complex 2 will come in because it increases the affinity to the promoter region, and it'll transcribe proteins that are pro-apoptotic, meaning that these are proteins that are required for the death of the cell. So there's a bunch of them, but I'm just gonna stick with 2 of them because I really like the name Puma, because one of the proteins named is Puma. The other one is Noxa. I'm gonna stick with the two just to keep it general. So Puma and Noxa are pro-apoptotic proteins, meaning they're made to destroy the cell, and it's P53 transcription factor that creates this protein. And what these Puma and Noxa are part of a very big superfamily of proteins called BCL2. So it's a BCL2 protein. These BCL proteins have 4 domains. 1, 2, 3, 4. 4 domains. That's the segmentation. That's what makes them all the same, is they have these same protein domains in them. They have these same amino acid sequences, the same throughout all of them. That's what puts them into a family. But this Puma and Noxa, which are pro-apoptotic, only have one of those protein domains. But it's enough to put them in that family of BCLA2. It's enough to put them in that family. And that protein domain is BH3 only proteins. We would call them BH3 only proteins. So it has the one of the domains called BH3. And what that's going to do is now I have to shift gears and go down to the mitochondria. So the mitochondria if we just want to rehash about the mitochondria has an inner cell membrane, which is more of like a prokaryote, and it has an outer cell membrane. And these are two separate cell membranes, two separate phospholipid bilayers. They're not like one that's split apart. They're two separate ones. And the inner one has these like little invaginations, these little U shapes, and they're called crista. The outer one is more like a round shape, right? And that's the outer cell wall of the outer membrane of the mitochondria. And in the outer membrane, there's another apoptotic protein that sits in the outer membrane, and that's called BAC. That's a pro-apoptotic. It's for cell death. It promotes cell death, and it sits in the outer membrane of the mitochondria. It's like an integral protein in the mitochondria. And out in the cytoplasm, there's another pro-apoptotic protein, and it's called BAX, B-A-X, and that sits in the cytoplasm. So they both have different starting points. And this, again, is in the same superfamily of BCL2. It's in the same family. This one has three of them. It has BH3, BH2, and BH1. It's only missing one of the amino acid sequences or the domains, and that's BH4. That's what makes it, that's the variation. That's the difference right there, is it has BH3, BH2, BH1. So anyway, their function, their role is they want to aggregate or join together, aggregate right at the outer cell membrane of the mitochondria, and they will form a channel that will release cytochrome C, which will start to cascade a series of events that will lead to apoptosis. So it's that release of cytochrome C. Cytochrome C is a key player in energy production when it's in the mitochondria. The interesting thing is when it leaves the mitochondria, it has a completely different function. And you're going to see that all throughout biology, is sometimes it's just the place that it's at. So anyway, BAC and BAX, again, which have two different starting points. BAC is in the outer membrane there. It's an integral protein within the mitochondria. They'll want to aggregate to make that channel so cytochrome C can come out and start a cascade, which will lead to captase enzymes and a cascade, which I'm going to get to in a different video. But for now, I'm just going to stick with this because this is a lot in one. Another BCL protein that's in this process is anti-apoptotic proteins, and that's BCL2 anti-apoptotic protein, BCL XL, and there's a lot of them, but I'm going to stick with two of them, just again to keep it general. So we'll stick with BCL XL and BCL2 anti-apoptotic protein. Now you might get confused there because the superfamily is called BCL2. The superfamily is called BCL2. One of the anti-apoptotic proteins is also called BCL2. So you're saying, what the fuck's going on? What happens is that was the first one that was found. So if it's the first thing that's found, they don't just simply change the name. What they did was they made that the name of the superfamily. So when you're identifying that specific anti-apoptotic protein, you're saying BCL2 anti-apoptotic protein. So you're identifying that single one, because if you just say BCL2, people are going to think of the entire superfamily. So you have to identify it as BCL2 anti-apoptotic protein, because again, they kept it as the same name because it was the first one that was found. And the anti-apoptotic proteins have all 4 protein domains. So those are the ones that have the whole thing, the whole package. It has everything. It has all the ones that the protein domains that'll group it into that superfamily. It has all 4 of them, and those, what their function is and what their job is to inhibit or block back and backs from aggregating and making that channel. And the reason is because if there was nothing to inhibit it, if there was nothing to stop backs and backs from doing their job, we would just fucking die, because our cells would be made, and then backs and backs would come in, and then the cell would die before it even finished the cell cycle. So without those anti-apoptotic proteins, back and backs would be left to their own business, and we would die. So we need something there to block it or inhibit it until it's necessary to kill the cell. So they block and block and block and block and block until it's necessary for the cell to be destroyed. For example, for a virus or bacteria or cancer, the cell will want to kill itself. Program cell death. It will want to die. So what the Puma and Noxa will do, which again are made by the P53 transcription factor, in a sense, it's made by that. What they'll do is they'll inhibit or block the inhibitor. They'll stop BCL2, anti-apoptotic protein, and BCL-XL. They'll block them. They'll stop their function. So BCL2, anti-apoptotic protein, and BCL-XL will no longer be available to block backs or back. So back and backs are left to their own business. There's nothing stopping them to aggregate at the mitochondrial outer cell membrane and letting cytochrome C out. And again, that'll form a cascade, which I'm going to get to in the next video. But I hope you enjoyed this one. And again, the last time I'm going to say it, me and Sasha Gray are friends, and that's it. Okay. I'll see you in the next video. Bye now.